So here I am, talking about the project that seems to have upset a few people, but I hope I can explain now exactly how all of this works. So, as a graduate student, long ago, okay, not that long ago, I'm not very old, but I wanted to understand how the Varroa mite works, because something was confusing me about this organism. We have a lot of references to it killing bees by removing a small amount of the bee's blood, but we don't have that next step that explains how that occurs. And that got me thinking about this subject and trying to understand it better. Allow me to begin. You are currently staring into the face of evil. If you're having a difficult time locating that face, don't worry, a lot of people do. It's this part right here. So Varroa destructor is a very strange organism. You can't always look at it and understand everything about it immediately. You have to look more closely. See, that face doesn't have any eyes. The mouth is difficult to discern. Where is the nose? It is not easy to always understand Varroa destructor, but you can if you pay very close attention. Now, this organism is considered to be the agent of primary concern in the trend that we have seen of honeybee health decline over the past several decades. It has caused a number of issues, and we haven't quite understood how it caused them, but we knew that something was wrong as soon as it arrived. Why? Within 10 years of its arrival in the United States, so it arrived in 1987, I had nothing to do with it. I was -2 at the time. But within 10 years of its arrival, it had killed nearly all of our unmanaged honeybees. So we knew that this parasite was different. It was different from the honeybee tracheal mites, which caused problems, but they didn't get rid of our wild bees. We knew that it was different from things like Nosema ceranae, which caused problems, but not to this extent. Now, in 1987, when this organism arrived in the US, and not long after it arrived in Germany, we knew that it was coming. But for some reason, we waited until it got to the US to do any real study of this organism. That left us kind of in the dark about some very important things. So allow me to give you a little bit of history. This organism was first described in 1904 from specimens collected in Java, Indonesia. And so this is the region of the world where it was originally native. You could find it Malaysia, Burma, Thailand, General Malay Archipelago, that whole region of the world. And then it started to spread. Why? Because it was able to jump from Apis cerana, the original host, to Apis mellifera. And Apis cerana is only found in Asia. But Apis mellifera has been able to move all around the world. So, as it moved outside of Asia, first it began moving in Asia. It was originally not found in China or India. It moved into China, it moved into India, it moved into Russia. And this is where things got interesting, because then it was an invasive species. So then people had a reason to study it. So the very first studies written about this creature are written in Russian and written in Chinese. That actually becomes very important as we go on, because this is really the crux of my study. What you see here is Varroa destructor on the underside of a bee, and up until the work that I conducted at the University of Maryland, we could not tell you what was happening here. Is this parasite feeding on the bee? Is this parasite feeding on the bee's blood? Pretty much every poster, publication, fact sheet that you read would tell you, prior to 2019, that Varroa destructor feeds exclusively on the blood of honeybees. But what I found, I was trying to find the original article that said that this occurs. I wanted to find the experiment that they conducted to determine this, and I, for almost a year, could not find that study. I searched far and wide, and eventually, finally got the paper. It was from the former Soviet Union in the late 1960s. And so this, or early 1970s actually, this was very interesting to me. I tried to read the paper. It was in Russian. I could not do so. I found a colleague from the Ukraine who actually translated the paper for me, and I found out that for years we have been saying that this parasite feeds on the bee's blood based on a mistranslation of a paper that almost no one read. The paper actually says in its concluding sentences that what the mite is feeding on seems to be a fluid. It's probably the bee's blood. More study will need to be conducted to determine the tissue that the mite is feeding on. But in the abstract, which was the only part of the paper translated into English, it seems to say clearly that the parasite is feeding on the bee's blood. But no one really read the paper. And so what we did was something that used to be a cardinal sin in science. People cited a citation of 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 a citation. And for that reason, no one had to read the original. That is not good, and we have to stop allowing that to happen. So I decided, since no one actually proved that this parasite feeds on the bee's blood, I should look into this, because maybe this is the reason why there are these 9 different pathologies that we see associated with Varroa, and we cannot fully attribute them to its feeding on the bee's blood. Something about it seems to be confusing. So I jumped in, but I was scared to jump in head first, because when you are a graduate student, you are judged by your ability to present an original contribution to the scientific community. And if you cannot, you have to go back to the drawing board again and again and again. I actually have a friend who was a graduate student in the same program for 11 years because he had to keep going back to the drawing board. So I decided I'm going to check and make sure that this is a worthwhile investment of my time. So I had 3 different expectations that I had if Varroa is feeding on the bee's blood. We call this the hemolymph in insects. If it is feeding on the hemolymph of the bees, these are the 3 things that I would expect to see. The first of these expectations is that the mite's digestive system and their excrement would show very close similarities to the digestive system and excrement of other creatures that feed on blood. Right? It seems fairly straightforward, correct? See, it actually takes a lot of work to feed on blood. You have to remodel your digestive system because blood is made mostly of water. And if you feed on something that is mostly water, you have to have a way to get rid of that water without swelling your body up so much that you pop. So, let's start with the excrement first, which is usually not something you hear in presentations, but this time we will. We will start with the excrement of the mites. Now, these white splotches that you see inside of the cell may look familiar to you. Some of you who have looked very closely at your brood frames will notice those white splats. Those are actually the communal toilet that the mites use. Every mite inside of the same cell will deposit their waste in that one spot. That made it easy for us as researchers to collect that waste and analyze it and determine what it is. You can tell a lot about what went into a creature based on what comes out of a creature. This allows us to determine what something was eating. But this is not what we expected to see with Varroa. It is feeding on we've been told that it is feeding on a food that is mostly water, but out of the back end, we find a very, very dry waste product. It's actually crystalline guanine. Now, guanine crystals typically result from the breakdown of organ meats. There's a lot of nitrogen in them. You typically do not find a lot of this kind of waste product if you're a creature that feeds on blood. Interesting, but still not enough to make any sort of definitive pronouncement. But another thing that I was interested in is the digestive system of the mites. You see, the digestive system is interesting because this is what the digestive system looks like for another type of a blood-feeding insect called a hemipteran. So if you have heard of kissing bugs or bed bugs, they have a digestive system that has to have something called a filter chamber. A filter chamber is a section of the digestive system that runs into the body. So this is the esophagus, this is the part that runs into the body. And it doubles around right here at the rectum, and it exchanges all the water from that blood into the rectum such that the creature can concentrate all the nutrients instead of just getting a meal made mostly of water. Any creature that feeds on blood or any really dilute fluid has to have a system like this, or they will just get a very diluted meal and will have to swell to ridiculous size. You can see an example here. Watch closely. On the back end of this tsetse fly, there is a fluid being deposited. This shows you how the filter chamber works. This tsetse fly was swelling up so much and so quickly that it had to allow its filter chamber to work, and so it pushes out the water from its food and a bunch of dissolved ions, and then begins to digest the blood from its meal. So if Varroa was feeding on the bee's blood, we would expect something similar. But you'll notice here, in this video provided by Dr. Alan Cohen, that the Varroa mite, and sorry, the leg of this drone that it is feeding on is a bit in the way, but this is the front end of the mite here, the back end of Varroa here, and you can actually see its digestive system working because its exoskeleton is just clear enough for you to see through it. And let me start it over one time, there you go. This is the rectum, and notice that no water is being deposited behind it, and notice that the mite is not swelling up. You can also notice that the food that is going through its digestive system is very white and very viscous. And this is a little hint. The blood of your bees is neither white nor viscous. Interesting. Okay, so I think I've made my point with this, and I will keep this moving. But this mite does not have a filter chamber. So that means it does not have the apparatus necessary to digest a meal that is composed primarily of water. So let's keep looking, because there may be more to this. I wanted to know, for the next of my expectations, is this mite closely related to other parasites that feed on the blood of their host? You can also learn a lot about a creature from what it is related to. Many creatures of very close relation share similar things that they feed on because it takes a lot of evolutionary time to change your digestive system. So let's look at what Varroa destructor is related to. Everyone, you are now seeing a phylogenetic tree. This is a genetic representation of the relatedness of a lot of different organisms. They are arachnids that you are looking at here. And so this list spans pretty much everything from the spiders and whiptail scorpions all the way down to the mites. This is where the mites begin. And right here at the mites, we see that there are some blood-feeding mites. Let me magnify this for you. There are some blood-feeding mites, the Argasis, that's a type of tick, the Ixodes ticks, another type of tick, and an Orthonisus. These are the types of chicken mites that feed on the blood of chickens. They feed on blood from the feathers. Now, we would expect that if Varroa is feeding on the bee's blood, that it would be related to some creatures similar to this. But instead, you find Varroa all the way down here. It is as distantly related from those creatures as it can be. Interestingly enough, let's look at what it is related to see what we can determine. So I'm sure you've already guessed this, but it's very closely related to the Cosmolaelaps, Lasiaceus, Macrocheles, and Gammacyphus mites. Of course, what else would it be related to? And please do remember those names. They will be on the exam at the end. Okay, all right. So, the Lasiaceus, Cosmolaelaps, Macrocheles, these mites and the Gammacyphus, they feed on their host through a process that we call extraoral digestion. Their mouth parts, as well as their digestive system, is structured so that they can stick their mouth into a creature, release digestive enzymes into that creature. Those enzymes will break that organism's organs down, and they can suck up that fluid that results from it. And we find that Varroa destructor's mouth parts are very similar to the mouth parts and to the digestive systems that you see in these creatures, which suggests that Varroa is not feeding on blood, but feeding on some of the bee's organ by extraoral digestion. So, as a graduate student, I thought, maybe this project could work. But first, I want to know one more thing before I jump into this project. I want to know where the parasites feed on the bees. So we have looked at two things now. You can learn a lot about what a creature eats from the way its digestive system works and from what comes out the back end of this creature. You can learn a lot about a creature from what it's related to. But you can also learn a lot about a parasite based solely on where that parasite feeds. Think about it. Mosquitoes and ticks, they can draw blood from you pretty much anywhere on your body. It's one of the worst things about mosquitoes. But why is it that they can do this? It is because the blood in your body is distributed throughout your body. But if mosquitoes only fed in one spot on your body, it might not be the blood that they're interested in, but maybe a tissue that is exclusive to just that spot. Well, let's see what kind of parasite Varroa is. For this, I had to conduct my own observational study, as there was no study of that sort already in the literature. And so I took several brood frames out of many colonies at the University of Maryland and at the United States Department of Agriculture. And with these colonies, I looked at the bees that were inside. I pulled them off the frame and looked at where the mite was on the body of the bee, where it was feeding on its host bee. And then I created this heat map so that you can understand what it is that was found in this study. And this was very interesting to me at the very beginning of this work. Take a closer look. Varroa destructor, more than 95% of the observations, these mites were found on the underside of the bee's body, between the plates that make up the abdomen of your bee. Why is that the case? Why do we not find them feeding here, where you can also find blood, or here, where most of the blood is concentrated? Because the thorax needs a lot of blood for the flight muscles. A large amount of blood goes through the thorax all the time. But anytime we found the mites on the thorax of the bee, and that's less than 5% of our observations, they were never feeding. They always had their front legs extended into the air and their mouth retracted into their face. This is something that we call questing phase. It is the same thing that a tick does when it hangs out on a blade of grass and waits for a deer or a mouse to walk by. It's trying to find a new host. Why is that important? Well, it's important because people are used to seeing this kind of image when they look for a picture of Varroa on a bee. And it has led many people to believe that this is the natural way that Varroa hangs out on the bee's body. But this is not. This bee is dead. Did anyone notice this? Did anyone notice that the bee has a mohawk? This bee has a mohawk because the wax was being used to slick this bee down and hold it in place, but the bee is dead. And so the mite is trying to get off of the bee's body. A lot of the pictures that we have on the internet of our bees, many of them are dead bees because they are a lot easier for you to get a good picture of a mite on their back. So here is why this is a problem. This is a big problem because when I would give presentations to beekeepers, and I would ask them, do you treat for Varroa? And the beekeepers would tell me, no, I don't treat for Varroa, but I don't treat because I don't have Varroa. Oh, okay. Tell me about your operation. How do you not have Varroa? And how do you know you don't have Varroa? And so I said, what is your way of monitoring to see your mites? Do you use the sugar shake? Do you use alcohol wash or soapy water? And they say, no. I inspect by eye. Pictures like this make it seem like Varroa is conspicuous. That if you just take a frame out of a colony and look at it, you will easily see a big red dot on your bees. But if you are not turning your bees over and looking at the underside of their body, you are unlikely to find Varroa. Because this is where they spend more than 95% of their time. If you are not looking at your bee's underbelly, you have not really seen Varroa in its most natural state. The time where Varroa climbs on to the thorax of the bee, as you can see this one here, is when there is what we would call in the US a real estate crisis. When so many of these mites have already attached to the bee that it is no longer a good food resource, when the bee is dying or dead, they want to climb onto the highest point on the bee's body so that it is easier to attach to another bee. They will also do this when the colony has been damaged so heavily that they are worried that the colony will die, and they want to go to a new colony. They will attach to the thorax of a forager. And the big problem in the US is that because so many beekeepers were looking at their frames and waiting to see the mites on the thorax of the bee, they were waiting until circumstances where the colony was already dying before they even treated it. And then many of them would tell me, I treated my colony last year and it still died. I'm never treating again. And I ask them when they treated, and they tell me when they saw a bunch of mites on the bee's thorax. So this is bad, and I'm glad that we are learning that this is not the natural state under most circumstances for the mite. But we also see here that Varroa has done a really good job of hiding itself. This is a good example. You can see this is one of the plates of the bee's body. It is called a sternite. This is another of the plates of the bee's body. And this is Varroa destructor in between. We have taken this using a fluorescent confocal microscope, and you can see that the mite is just stuck in between the plates that make up the bee's body. It is blurring the line between an internal and external parasite. That means it is very difficult for us to see things like feeding. But let's get to that in a moment. All of that to say, the first three of my expectations were not met. All three of my expectations were not met. And so it is actually unlikely that Varroa is feeding on the bee's blood. So I thought, all right, maybe this is a good project. I will continue studying this. But before I could get to that, some researchers, I gave a presentation to my department, and some researchers stopped me and they said, no. There's nothing to see here. Well, I think there is. They said, no, because Varroa is a phoretic on the adult bees. So what it is doing on the body of the adult bee does not matter, because the word phoretic in ecology, in biology, it specifically means that this creature uses another creature as a mode of transit to get from one place to the next. But it values this creature so much as a mode of transit that it does not feed on this creature. I was told that Varroa only feeds on the brood. On the adults, it fully suspends feeding. And so the work that you've done so far was fun, but it doesn't show anything. I did not think that this was true. But they brought a very, very, very important piece of information to me. They said, Sammy, people have been studying this parasite since 1904, for more than 100 years. And in all this time, studying this creature, never even once has anyone seen any physical evidence that Varroa is feeding on the adult bees. No bites, no wounds, nothing. Now, we have many studies that can show you the entire wound site from when this creature feeds on the brood, but nothing for the adult bees. And with this much negative data, that must mean something. That must mean it is not feeding on the adult bees. But I did not think that that was the case. I thought the brood is very easy to see. It doesn't have any plates that can hide the mite's body like the adult bees do. And maybe if we could look under this plate, we could see what this mite is doing. But then I learned why no one's been successful at doing this before. Because the bees don't like it when you pull their plates up and down. Not very fond of this. So I had to find a way to stop the bees from fighting me in this process. So I took the bees and the mites, and I put them in liquid nitrogen. This froze them. It's negative 197 degrees. It's very, very, very cold. It froze them both solid. So this is an image from an electron micrograph. This is one of the plates. This is a sternite. This is another sternite that the mite is under. And this is Varroa destructor, jutting out from behind it. And what I want to see is what's happening underneath this plate. And so, very carefully, I broke the plate off, pulled the mite out, and this is what we saw. Actually, it's very difficult to see without adding the color back, so let me... Okay. So now that we have the color for this image, you can see two very interesting things. This object here and this object here. Both of them are the same thing. They are actually the foot pads of the mite, the ambulacra, as we call them in the lab. The mite was so well embedded into the host's membrane that its foot pads remained behind even after we pulled the mite out. That was very, very lucky because it allowed us to see something very clearly. If you follow the indentation of the legs, the front two legs always end at the mouth. But instead of seeing the indentation of the mite's mouth, we see something interesting. We see a hole. And so we colorize this image so that you can see that the shape of the hole is the exact shape of the mite's mouth parts. This is the bottom of the mouth. This is the top of the mouth. You can see the top of the hole here, the bottom, which presses it out. And it's the exact same size as the mite's mouth. So we knew it was exactly what caused this hole. But they told me to stop again. They said, Sammy, keep in mind, I know what you found here looks really exciting, but it's very possible that the mite isn't eating the bee, but that it just wedged itself in there so tightly that its mouth just popped through. But maybe it's not eating anything. You have not shown that it is eating anything. And you know what? I don't want to make fun of these researchers for their skepticism. Skepticism is essential to science. They made me a better researcher because I was trying to tell the world that we have been spending more than half a century saying the wrong thing about this creature. And that a lot of what we have done has been based on assumption, on top of assumption, on top of assumption. So in order to show this, I can't assume anything. So instead of just saying, because there is a hole in the bee's skin where the mite's mouth was, I can't say that the mite was definitely feeding from just that image. I had to start conducting some light microscopy as well. Now, I should say that all of this microscopy is a result of the wonderful help of the United States Department of Agriculture. They have these gigantic microscopes all in one big facility. They are millions of dollars of microscopes. They are terrifying, but they let me use them as a graduate student and taught me how, and I am so grateful to them because now you can see what I saw. Because this is the image, one of the images, that made everything so clear to me. Check this out. Now, first we have Varroa. This is Varroa destructor. We have done something called a thin section, where we have used an ultra microtome to cut all the way through the bee and all the way through the mite at the same time. And so you can see the mite's body here. This is the bee. This is one of the plates, one sternite. This is another plate, another sternite. And this is the inside of the bee's body, separated from the mite by this one membrane. You can see there's a hole going through this membrane right here. And the fascinating thing is that when there is no mite in this region of the bee's body, this interesting tissue right here runs all the way under the membrane and forms a very thick, dense layer. But when the mite is present, whole sections of this tissue are missing. And the longer the mite is present, the more of this tissue is missing. If we get closer, because I really wanted to know what this tissue was, I kept inspecting, and every time I would see this stuff, this goo, and I was not sure what it was. I started calling it the schmutz. Now, this stuff is actually the internal contents of honeybee cells, specifically fat body cells. The fat body is the liver of the bee. And when you have a cell, the insides of that cell are not supposed to be outside of the cell unless something very bad happened. And we were able to see this because this one fat body cell remained attached to the membrane. And we could look at what was inside of this cell and determine that it was the same stuff we were seeing out here. The only time you see cells just open like this is when something like extraoral digestion is occurring. When when enzymes have been spit into another creature's body, and those enzymes break its cells down into a fluid that is composed of the internal contents of its cells. So this strongly suggested to us that the mites are feeding on the bee's fat body, on their liver, through extraoral digestion. But that wasn't the end. I had to keep going. We zoomed in even closer. This is the same image. We've gotten closer and closer and closer, and now we see something else that is very interesting. What you'll see here, if I can, I'm standing in front of it, I see. There are two colonies of morphologically distinct bacteria just on the other side of the wound site. And what you would normally see here is that when bacteria invade the body of a creature like a bee, the immune system starts fighting them immediately. But what controls the bee's immune system? It's actually the fat body. And so it is very much expected that if you damage the fat body, then bacteria and fungi and viruses can get into the bee's body, and they can fight much better than the bee can because Varroa has been destroying the tissue that controls the bee's immune system. Let's keep going. This picture is actual. You know what? We're going to back up 1 second. Because normally, when I put up that picture, people go, ooh, ah. And so I think you guys missed your cue. So we're gonna go back. We're going to do this one more time. There you go. Thank you. I appreciate that. Yep. It feeds my soul. It really does. Because this image took forever for us to get it. It's 66 tries? So yes, we froze a bee in liquid nitrogen, and then I used a very, very thin blade called a feather blade to cut through the bee and the mite's body so that it would crack open like a clamshell. It just fractures open. And then you can take pictures of both sides. And what you see here is that the bee, let's see, so this is once again the sternite of the bee. The hair is coming off of it. This section right here is the intersegmental membrane. This is the membrane between the plates. And just on the other side of this membrane is a big deposit of fat body. You can see some of it has even been hollowed out by the feeding of the mite. And there's plenty more fat body under it. And then you can even see the digestive system of the mite in here with something that looks very similar. Okay, so I'm just going to leave it here for 2 more seconds. Thank you. But I knew that this would not be enough to convince everyone. So I wanted to conduct some experiments to make it very clear what is happening here. People were very quick to point out that this fat body tissue, and this is what fat body looks like. Many beekeepers have told me, I've never seen fat body before. I disagree. If you've been keeping bees for any amount of time, you have seen a lot of fat body. Have you ever seen your brood? Because the skin of your brood is transparent, and all of that white stuff is fat body. And inside of the bee's body, as an adult, if you cut open the abdomen, this is the abdomen of your bee, if you fold it open like a clamshell and remove the digestive system, this is the largest distributed organ in your bee's body. All of that is fat body, all of that white, gooey matter. And it is not just the body fat of your bee. It is very important to your bee's development. And what we've found in our study is that Varroa only feeds near this tissue. This is the inside of the abdomen, and notice in our study we found that they would only feed in this region of the bee's body. Now, this is where things got really fun, because I got to use glowing fluids to solve a problem. This is what every scientist dreams of doing, I promise you. So, this was really fun because the sugar solution of the bees is a really good way to get something into the bee's body. Bees will feed on pretty much anything with sugar in it. They will ask no questions as long as it is sweet. And so I wanted to color, to mark the organs of the bee's body, different colors, and then look at the digestive system of the mites. And depending on what color the digestive system is, I can tell you what that mite was eating. So one of these that you see right here looks kind of pinkish. This one is called Nile red. It is a chemical that is very lipophilic. It will attach to fatty organs, like the fat body, and will remain attached to them. And it will only glow when attached to the fats. And then we have uranine O, which you can see right here, the yellow one, in the sugar solution. This uranine O has the opposite biochemistry. It will attach to the blood of the bee, and it will remain there, and it will glow when it is in the blood. I love chemistry. Let's keep going for just a moment. This is a bee that has been fed both of those chemicals in its sugar solution for 5 days. You are looking at the abdomen of this bee, and you can see that it is glowing quite brightly in both yellow and red. The yellow is the blood of the bee. This is a sample that we have removed from this bee, put on a slide and put under the microscope. So this is your lights on, lights off image of what you're seeing here. And then red for the deposit of fat body that you see right here, that we have removed from this same bee. Now here is the part that I want you to pay attention to. If Varroa is feeding on the bee's blood, then Varroa will glow yellow. If Varroa is feeding on the bee's fat body, then Varroa will glow red. Oh, wait. There you go. So this is the inside of Varroa destructor's body. You can see that the digestive system is glowing so brightly that you almost can't even see the mite. And we know that it was the fat body because we got very close images of what the fat body looks like when it has been stained with Nile red. This chemical, Nile red, will color all of the cells of the fat body bright red. These cells, called adipocytes, are the fat body cells, they're primary cells, and they have absorbed all of that red coloration and they are glowing. This is an image of that. And you can find this same fluorescence in the digestive system of mites that have been exposed to these bees. We did this more than 60 times to make sure that it was constantly repeatable, and every single time we only saw the fluorescence from the fat body. But because I am trying to tell people that they have been wrong about something for more than half a century, I had to be very thorough. So I also did tests where I used a confocal microscope to get even closer, just to make sure that this was coming from the digestive system of the mites. You can see the entire shape of the mite's digestive system just in the fat body tissue that is consumed. All of those glowing red dots are from the fat body. But then we wanted to know, what happens if there is no fat body biostain? What happens if you don't give them Nile red? What if the Nile red is glowing so brightly that it is obscuring the fluorescence from the blood? Maybe the mites are eating the blood and the fat body, and that would show us. So I fed some bees only a sugar solution that had no fluorescence in it. You can see that the mites, some of their muscular tissue glows a little bit green, but you're looking for yellow, and we see no yellow coloration in the digestive system of these mites because they were only fed sugar water. Now the question is, if we feed them from bees that have only been fed the biostain for the bee's blood, will they glow at least a little bit? Or will they look just like this control mite? These are mites that were fed on bees whose blood was colored with this biostain. They look exactly like the control mites that were only fed from bees that were given sugar water. The mites are not feeding on the bee's blood. But this is a point where beekeepers ask me, why does this matter? If the bees are being fed on by this parasite, should I care if they are eating the bee's blood or if they are eating the bee's liver? Either way, they are eating my bees. That is a point, but I'm not sure that it is a good one. Consider this. Think about a mosquito landing on you. It feeds on some of your blood, and then it flies away. You might get a little itchy, but you will be okay, unless maybe it has a virus or something. But imagine the mosquito lands on you, liquefies part of your liver, and sucks that out of your body. You will be more concerned, I think. That is because the blood inside of your body is constantly renewed. But it is very difficult to grow a new liver. The liver inside of the bees has 9 essential functions that it conducts for the bees. And this is why I wish that we did not call it the fat body, because a lot of people hear this word and they think it just means body fat. But that is not correct. This is an entire organ that does many important things. I have a list for you here of the 9 different functions of the fat body, and I want you to know that every single one of these has a pathology that is related to it. I told you at the beginning of this talk that there are 9 pathologies that we know of that are associated with Varroa feeding on bees. And we have never been able to link all of those pathologies, all of those diseases, to the feeding of the bee's blood. Well, I went through and I read all of the papers that detail these things. So the fat body is what controls energy storage and metabolism for the bees. It allows them to take energy that is in their body and release it into their flight muscles so that they can fly to some flowers and then fly back home. But when Varroa has fed on bees, oftentimes they will only be able to regulate enough energy to fly to the flowers, but not enough for the return flight home. And some of the bees will die there. The paper that describes this says, we do not understand how the removal of, or we are not sure how the removal of a small amount of the bee's blood would cause this specific pathology. But what does make sense is that because the fat body controls the bee's metabolism, that if you damage or eat the fat body, you will damage its metabolism. So what seems like a pathology has come from the disruption of the normal and healthy functioning of the fat body. It is the same for pesticide detoxification. When Varroa has fed on the bees, for some reason, when you expose them to pesticides, they die a lot more easily, even if you expose them to an amount of a pesticide that was considered to be a sublethal dose. The tissue that breaks down pesticides such that they do not kill your bees is the fat body. The tissue that makes the proteins that the bee's body is made out of is the fat body. It's also the tissue that stores them. Another study detailing this says we don't understand how the removal of a small amount of the bee's blood would cause them to lose so much protein. There was less protein in the amount of blood removed than the bees are lacking now. Another very important study looked at immune function in the bees, and it showed that the immune system of the bees is heavily depressed by Varroa feeding on bees. That means that fungi, bacteria, and viruses can more easily overcome the bees when they have been fed on by Varroa. This paper had a very difficult time being accepted in the scientific community. There were many who still would not accept this paper because they did not understand how just taking out some of the bees' blood would cause their immune system to drop so much. But because the fat body creates the peptides that build the immune system of the bees, you would expect that damaging the fat body would damage the immune system. So every single one of these functions of the bee's body, including temperature regulation, which is important during the winter, that is damaged when Varroa feeds on the bees. We now understand that because the fat body regulates the bee's body temperature. We now understand that the fat body regulates the bee's water loss, such that the wax gland is part of the fat body. It produces wax that coats the outside of the bee's body and keeps the bee's water from evaporating out of their body. And when they are damaged by Varroa, for some reason, their cuticle, the wax over their body, is much thinner than in bees that have not been fed on by Varroa, and water evaporates out of their body, and they dry up and die much more quickly. Every single one of the functions of the fat body is impacted by the feeding of Varroa. And this provides us with a unified theory to understand why there are so many seemingly unrelated diseases that come from Varroa feeding on bees. All of it is not the viruses. Even when Varroa is naive to viruses, and we have done this in the lab in Beltsville, in the United States Department of Agriculture's bee lab, even when Varroa has no viruses, it still causes a lot of damage when it feeds on bees. Now, with the viruses, things get even worse because the bee's immune system is depressed. But it causes a lot of damage because it feeds on this tissue. Now, I knew that in trying to get this work published, that people would tell me, I'm not sure I believe this because you have conducted this work on adult bees. And when Varroa feed, a lot of their feeding happens on the brood, and maybe in the brood, that is where they feed on the bee's blood. And so in order to show very clearly that Varroa is feeding on the bee's fat body, both as the adult and as the juvenile bees, I had to do something that my advisor was very nervous about. I told my graduate school advisor that I want to create a system where I can feed the mites what I want to feed the mites, because that's the only way for me to tell what it is that they need in their diet. So if I feed fat body to these mites and they cannot survive and they cannot produce eggs, then we know for sure that they are not eating the fat body. If I feed them the blood of the bees, and they survive and they produce eggs, then the blood is what they were after, at least in the immature bees. But the reason why why my advisor was very concerned with me doing this study is because many researchers have attempted this for a long time. It is very difficult to get Varroa destructor to survive outside of the honeybee colony, and no one has been able to get them to lay eggs. And I thought maybe the reason for this is because everyone has been trying to feed them something they do not want to eat. So I decided to tackle this. I would feed them a diet that is composed entirely of the bee's blood, or entirely of fat body, or of some combination of the two of these things. So, the very first thing that I had to do for this study was create a decoy bee. Hopefully, some of you were fooled by this image, but one of these things is not like the other. One of these things is actually a pill. This is a size 5 gelatin capsule, and it has fooled this mite into believing that it is a bee. It is the same size as a bee, same shape, similar to this pupa that you see here, but it does not feel like a bee. So remember that Varroa cannot see. And so the most important things to it are touch, taste, and smell. And so for this study, I took this wax called parafilm, and I stretched it very thin, such that it was as thin as the skin of these developing bees. Very, 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 very thin. 15 microns thin. And I wrapped it around the pill. And nothing happened. The mites would not feed. They would not pay any attention to it. I am very embarrassed that it took me so long to figure out what the problem was. It does not matter if it just looks like a bee, if it just feels like a bee. It has to smell and taste like a bee. So I started taking the parafilm and rubbing it against the bee's body, against the brood, before wrapping it around the pill. And then my mites would feed. It took me too long to figure that out. I'm a little embarrassed. But we cut a trough into the bottom of this pill, stretched the very thin parafilm along that trough, and then injected the food into that pill. So I made a small hole and injected the hemolymph of the bee, or injected fat body that I had blended into that cell, or a combination of the two of them. And then we closed the lid with cling wrap and put them inside of an incubator to watch and see what happened. And this is where things got interesting. Does anybody want to guess what this object is that was on the cling wrap of one of these cells? Any ideas? Oh, right. Did you say the egg? Oh, okay. Well, look at you guys. All right. I'm impressed. So I personally thought, this is an egg. This is the coolest thing I have ever seen. I want to backflip down the hallways because no one has been able to get these parasites to produce eggs. And this was in the fat body treatment. But I wanted to wait until I could get it into the lab because it was 4 o'clock in the morning, and I was not sure that I wasn't delirious and hallucinating. So I took it to the electron microscopy lab, we put it in the microscope, and surely enough, it is an egg. And the mites produce these eggs when you feed them fat body. I was so excited, I even named this egg. I named it PhD, because this very clearly to me meant that they were going to give me my degree, and I was very excited. But, oh, thank you. Thank you. So, I want you to pay attention to this graph, because when Varroa is fed no food at all, when it is starved, it does not produce eggs. This should surprise no one. But this is the part that's surprising. If you feed a mite 100% hemolymph, it produces no difference in the amount of eggs from when it is starved to when it is fed hemolymph. if you feed them fat body, and only a little bit of fat body, starting at 25% fat body to hemolymph, up to 50, up to 100%, they will start laying eggs at 25%, and they will lay eggs most regularly, like under more natural circumstances, if you feed them 100% fat body. That is when they really start laying eggs. No blood required. But let's also look at the survivorship. How well do the mites survive when they are fed fat body or when they are fed blood? What you see here is called a survivorship curve. It shows you how easily creatures die when you put them in different treatments. You will notice that this line right here, this black line, goes over and drops all the way down. This is a whole set of mites that died at the beginning of the study. These mites were starved. You can see that there is a similar line where all of them are dead at 48 hours. The ones that were starved and the ones that were fed the bees' blood were the only ones to die that early in the study. It seemed like they weren't getting enough nutrition, which is not surprising because the bees' blood is mostly water. Very few creatures can survive only feeding on the blood of an insect. The blood of a human has a hematocrit, that is the amount of cells that are in this, that is nearly 10 times greater than the amount of cells and the amount of nutrients that you find in the same amount of a bee's blood. It is very nutrient-poor food. But what you will find is that the only mites that survived the full 7-day duration of this study were the ones that were fed fat body. The ones fed 100% fat body survived the best, but even if they were only fed 50% fat body, they could survive until the end of the study. Now you might be wondering what happened in this part of the graph, because we have a stepwise system of growth going from the amount of fat body you feed to how many eggs the mites are producing. But for some reason, when you get to 75% fat body to 25% hemolymph, there is a drop-off. This was very interesting. It was actually really sad at first, but then really interesting. The mites, food, if you mix together 75% fat body and 25% hemolymph, you get something that we call a growth medium. And a growth medium is something that grows bacteria or fungi very, very quickly. And the fungi that started growing on this 75% to 25% fat body was extremely virulent to the mites. It killed them in just hours. And then it started growing a whole fungus mat out of the mite's body. And it was really, really exciting. So we started talking with people about this to see if maybe we could turn this into some sort of treatment for the mites. And there are still people working on this, but it is unfortunate that bees use a lot of propolis in their colonies. And even though we could get this fungus to kill the mites in the lab, when we tried to get it into the colony, the bees removed all of the fungal spores before they could germinate. The bees removed any of the fruiting bodies that did germinate, and they rubbed propolis all over everything, and it has an antifungal and antimicrobial effect. So it doesn't work well inside of the colony, but still really cool, right? Yeah? Okay. Oh, thank you. So this work was finally published with the Proceedings of the National Academy of Sciences, which I was very excited about, because when you're working on this kind of research, and it takes years and years and years to get it out there, you want to get it to as many people as possible. And so we had to pay nearly $4,000 extra for the publication, but I wanted it to be available to everyone. And so because we paid on the front end, any of you can read the paper and see any of the images, and you can even use the images that you have seen in this presentation, because this paper was published open access. And you have the University of Maryland and Dennis Van Ingelstorp to really thank for that, because he foot the bill. I did not have an extra $4,000 lying around. So I'm very grateful for that. And if you want to see more of this, you can find it. Just Google Varroa fat body. It is the very first thing that will come up. Now, in conclusion to this, there are 3 things that I want you to think of as very important to take away. First of all, when you choose to treat is just as important as what you choose to treat with. If you're a beekeeper, you do not have the impressive resistant stock that Jeff was talking about earlier, and Dr. C were talking about, well, then you're probably going to have to treat your bees. So remember to treat them at the right time. We've been telling people, treat your colonies in the fall, right before the winter, so that there are fewer mites going into the winter with your bees. That makes sense. But that doesn't take into account that the mites are feeding on the fat body. If they were feeding on the bee's blood, that's not as big a deal, because the blood is going to be replenished. But the fat body that they emerge from under the cell capping with is all the fat body they will have for the rest of their lives. It will not be regrown. And so if you allow your winter bees to be damaged by these mites, those bees will go into the winter and they probably will not survive. So it is important that you treat those bees in the late summer, early fall, when your winter bees are being produced. These are bees that have a different physiology than your regular bees. They have a huge amount of fat body. They need this fat body because the fat body has a protein in it that is called vitellogenin. And vitellogenin is able to extend the lifespan of your bees by reducing oxidative stress. So the more vitellogenin your bees have, the more fat body your bees have, the more vitellogenin they have. And the more vitellogenin they have, the longer they can live, so that if you have a long winter, they can survive the whole thing. This is the reason why you have to make sure that you don't just reduce the number of mites going into the winter with your bees. You have to reduce the number of mites that are feeding on your bees while they are under the cell capping and being produced for the winter, or they will not be healthy enough to get through the winter. In addition, it's important to consider, there was a study from the early 2000s that showed that when Varroa feed on bees, when they are fed protein, they cannot absorb it, or they cannot absorb much of it. And it was a very interesting study, but once again, no one understood how the removal of a small amount of the bee's blood would stop them from being able to store protein. Well, what is the tissue where the protein is stored? Oh, don't be afraid, it's the fat body. Don't worry, I'm not going to trick you. This is like Sunday school, where the answer to all of the questions is Jesus. Like, this is like that, but instead with fat body, all right? Okay, so yes. So, right. So, the tissue that stores the proteins in the bee is the, there we go. All right, thank you. Yes. And so, if you feed your bees protein, but they do not have the fat body to absorb this protein, it cannot be collected by them, and you have given your bees expensive poop. But even worse than that, if they eat too much of this protein and cannot absorb it, it builds up in their blood and it kills them. So if you have a colony that has very high mite levels, do not feed them protein thinking that the protein will help your bees. It will not. It can hurt your bees. Wait until another generation, so treat them, get rid of the mites, and wait until another generation has been reared, and then give them protein. And finally, there are a couple of rather large companies that are interested in using the information from this study to engineer a toxin that will kill the mites into the fat body of the bees. Now, I am already well aware there are a lot of people that this makes very nervous. For some people, they are very excited and they say anything that will help us fight the mites is wonderful. Other people are like, ooh, we do not want bioengineered bees or bees that are eating something that's storing in their fat body. But just so that you know, this is something that is being considered in the future as a way that we can potentially help the bees. But we have to look very closely at this. Because if you try to get something into the bee's blood, a systemic, that will kill the mites in their blood, that would be fine because the blood is constantly renewed. You get new blood, but you don't get new fat body. So if you end up storing a ton of this inside of your fat body, it could potentially biomagnify, and that could be bad for the bees and the colonies. So any work done on this has to be very heavily scrutinized to know for sure that it is not causing problems for the bees. And I think I'm getting close to time. Okay, cool. Well then, I would like to thank all of you for listening to this presentation. Thank you very much. Oh. Aw. Thank you. Thank you very, very much. Thank you.